I am Sergio Hernandez Torralba. I am a PhD student at Politecnica University of Valencia, and I am going to talk about my study based on the effect of softening pumpkins using macerating enzymes on sugar content. I will begin by introducing the current issues and the need for the development and the optimization of texture-modified foods. This need arises from the attempt to ensure the health and well-being of people with chewing and swallowing problems, a problem that mostly affects the aging population. In this sense, it's important to highlight that texture-modified foods are normally purs, cream, or subs, and unfortunately, these products may not be appetizing to some consumers. This lack of appetite, joined with the fact that many products have to be removed from the diet because produce discomfort to them when consuming, provokes eating disorder and unbalanced diets. Pumpkin was selected because of its multiple nutritional benefits, since these have a great vitamin richness, among which it can highlight its content in carotenoids as beta-carotene, minerals, and antioxidant capacity. Pumpkins were cut into cylinders with 2 centimeters of diameter and height and were immersed into 0.5 enzyme solution. Commercial enzyme with polygalacturonase, pectin methylsterase, and pectin liase activities was used. Then a vacuum pulse was applied for 5 minutes. After that, restoration to atmosphere pressure was carried out in 1.5 minutes to complete the impregnation process. To improve enzyme reaction, the samples were transferred to an oven at 36 Celsius degrees for an hour. Finally, the enzyme-treated samples were steaming for 5 minutes to stop the reaction. Parallelly, another pumpkins were traditionally cooked by means of steaming for 15 minutes. At the end of the treatment, all samples were centrifugated and the supernatant was collected and kept in frozen until the analysis. The analysis consists of measuring texture by compression assay and sugar content. Reducing sugars were analyzed using dinitrosalicylic DNase method, and total sugars were determined by phenylsulfuric acid spectrophotometric method. The maximum force of the compression assay was expressed as hardness. At figure 1, it can be seen the hardness of control, cooked, and enzyme-treated samples with standard deviation and different letters indicating significant difference by Fisher's tests. Enzyme samples had a hardness of 1.2 newtons, significantly lower hardness than control and cooked samples. Moreover, as shown in figure 2, enzyme-treated samples had a significantly higher concentration of reducing sugars than cooked samples. On the other hand, the enzyme-treated samples and cooked samples did not show a statistically significant difference, although as can be seen in figure 3, the enzyme-treated samples showed a slightly higher concentration of total sugars. This suggests that enzyme-treated samples contain a higher proportion of reducing sugars than cooked samples. The increase in reducing sugars for the enzyme-treated samples is due to the enzyme hydrolysis, as demonstrated by a higher percentage of hydrolysis calculated following the equation shown at the bottom of the screen. To conclude, I would like to highlight that the overall results demonstrate the capability of the softening technique mediated by macerating enzymes combined to a vacuum pulse for achieving suitable textures for aging population. Thus, texture-modified product has successfully designed enzyme-treated samples had a 1.2 plus or 0.5 newtons of hardness. These scores were 5 times softer than the cooked samples and 24 times softer than the control samples. Moreover, enzyme-treated samples had a significantly higher concentration of reducing sugars than cooked samples, and a higher concentration of total sugars but not significantly. This aspect could be an advantage for aging population who are prone to suffer caloric deficits due to their trend to decrease their food intake as a consequence of physiological changes and reduced appetite. However, further studies on the monosaccharides composition are needed to develop products for specific populations. I conclude my oral participation and give way to the rest of the participants of the Congress.